Transmission electron microscope, it is also called as TEM. In transmission electron microscope, use a beam of electron to create an image of the internal structure of thin sample such as molecule and tissue section. That means here we will observe the internal structure of thin sample of molecules and tissue sections. The electron beam will pass through the thin sample and creating a projection of image in the computer. Here you can see this is the transmission electron microscope column or chamber or tube, and after that the projected image can be observed in this computer. And in this transmission electron microscope will magnify nanometer structure up to 50 million times when comparing to the other microscope. The working principle of transmission electron microscope, and this is the tube of transmission electron microscope. This tube is in vacuum. That means there is no air is present inside this tube. And this tube is again divided into illumination system and image formation area and image magnifying area. So this is the electron source. This electron from the electron source, electron will be produced. So this electron beam will reaches to the condenser. The condenser will narrow the diameter of this electron beams. And also this condenser and this objective lens will focus and project the electron beams to the sample. And the presence of this objective lens and the objective aperture will enhance the magnification of the transmitted image. And this is the intermediate lens, and intermediate lens will also enhance or enlarged image when these beams, transmitted beams reaches to the projector lens. This projector lens will project the image into this fluorescent screen, and it is connected with the computer. So the projected image can be observed in the computer monitor. And here we told that it is transmission electron microscope. And in this transmission electron microscope, the electron light or beam is passed through the transmitted to the sample. For the transmission of the sample, before keeping the sample into the sample area, we have to do, there are some processes are there to making the thinner sample. The structure of the sample can be observed like crystal structure morphology. So with the help of this transmission electron microscope, we can clearly observe the inner structure of the smear or the sample or the cell or bacteria or virus, whatever it is. And the resolution is less than 50 picometer. 1 picometer is equal to 1000 nanometer. So you can imagine how much is the 50 picometer. And it provides 2-dimension projection in the sample. The sample preparation is complex when compared to the scanning electron microscope. And the sample must be thin, generally below 150 nanometer. It accelerates voltage range of 60 to 300 kV. It magnifies more than 50 million times. And a very small field or small part of the image can be seen through this transmission electron microscope when we are comparing with the scanning electron microscope.
Subscribe to Simply Micro Med. If you're curious about medical microbiology, infectious diseases, and unraveling the mysteries of tiny organisms, hit that subscribe button. Join our community of science enthusiasts, and let's explore the microscopic world together. Remember, knowledge is contagious. Spread it by liking and subscribing. Thank you.